A very good afternoon to everyone. Today we will study about meiosis. The term meiosis was given by Farmer and Murray. Meiosis include one round of DNA replication and two rounds of chromosomal segregation. In sexually reproducing organisms who are diploid, this meiosis process occurs. Meiosis has two parts, meiosis first and meiosis second. Meiosis first is the reductional division, where there is a separation of two homologous pair and the daughter cells has single homologous chromosome, while the meiosis 2 is the equational division, and here the segregation of sister chromatids occur and the daughter cells have only one sister chromatid, and this is the second one. Meiosis 1 has four phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. Meiosis first is called as the reductional division, where the number of chromosome get half after the division from the mother cell and the daughter cell have 1/2 number of chromosome. Prophase of meiosis 1 has further 5 sub-stages. The first one is Leptotene, second is Zygotene, third sub-stage of prophase 1 is Pachyten, fourth sub-stage is Diplotene and fifth is Diakinesis. All these 5 sub-stages of prophase 1 are very important and they refer to the crossing over in the non-sister chromatids of homologous chromosomes and then condensation. The Leptotene is the first stage, and here the chromosomes get replicated and they form two sister chromatids. These all homologous chromosomes are attached to the inner nuclear envelope and they form a bucket stage. This bucket stage facilitates the chromosomal pairing and synapses for the further zygotene stage. The second stage is the zygotene stage, and in zygotene the synapses of two homologous chromosomes occur. The synapses forms a bivalent or tetrad, and this tetrad structure is a very complex structure having a number of proteins. This synaptonymal complex has a central region and it is made up of lateral element, central element and transverse element. All these elements have a synaptonymal complex protein and these together form the central region of this homologous pair. The lateral element is made up of synaptonymal complex protein 2 and synaptonymal complex protein 3, SYC P2 and P3. The central element is made up of SYC P1 or synaptonymal complex protein 1. The transverse element is made up of synaptonymal complex protein E1, E2 and E3. These transverse, central and lateral elements together form the synaptonymal complex, and this help in the pairing of homologous chromosomes together and further help in the crossing over between them. The third sub-stage is Pachyten. In Pachyten, the crossing over occurs. During crossing over, a new protein RAD51 while REC-A protein in eukaryotes comes and bind to this synaptonymal complex and facilitate the crossing over. Crossing over is nothing but the chromosomal exchange between two non-sister chromatids of two homologous chromosomes. This is just the reciprocal exchange of chromosomal segment between the chromosomes. But 2 points has to be remembered always for Pachyten stage, that in male Drosophila and female silkworm crossing over never happens, and the second, a chromosome always has two reasons for crossing over. First is the cold one, where there is low frequency of chromosomal crossing over, and the second is the hot spot or the hot region, where there is a high frequency of occurrence of crossing over. The fourth sub-stage of prophase 1 is Diplotene. in diplotene, desynapsis occur or the dissolution of synaptonymal complex occurs except at one position that forms a X structure called as the chiasmata, where the two non-sister chromatids of two homologous chromosomes are attached, and this is called as chiasmata. The oocytes or the developing eggs of mammals are arrested in diplotene stage for years because the chromosomes are decondensed here and they are highly involved in RNA synthesis and they provide storage material for its development. Further, in amphibians, these chromosomes in diplotene stage form a lamb-brush chromosome which has a brush-like structure and they are highly involved in RNA synthesis. The fifth sub-stage of prophase 1 is diakinesis. During diakinesis, the chromosome gets condensed and RNA synthesis ceases. The condensed chromosomes form a pair and they are called as bivalent or the tetrad structure. The bivalent structure has 4 sister chromatids while the single homologous chromosome has 2 sister chromatids. These chromosomes leave the attachment of the inner nuclear envelope and they are arranged in the central part of the nucleus. The second phase is metaphase first. During metaphase first, the nuclear envelope get dissolved and the condensed chromosomes are attached at the metaphase plate. The attachment of homologous pair of chromosomes at the metaphase plate occurs by the attachment of kinetochore microtubule to these bivalents. The third phase is anaphase 1. Anaphase 1 is a phase where the two homologous chromosomes of a bivalent get separated while the two sister chromatids of one homologous chromosome remains together, and it happens only because of the presence of a special protein called as cohesine or the REC8 protein. This anaphase is different from the anaphase of mitosis, where from a mother cell the two daughter cells are formed having only one sister chromatids. Telophase is the last phase of meiosis 1. In telophase, nuclear envelope is again formed and the chromosomes get decondensed. During telophase, cytokinesis occurs and two daughter cells are formed having a pair of homologous chromosomes. Meiosis has two parts, meiosis 1 and meiosis 2. Meiosis 1 is reductional division and meiosis 2 is the equational division. In between meiosis 1 and 2, there is a small or shorter phase that is called interkinesis. It's a time gap between meiosis 1 and 2, and this is a very shorter, or sometime in cell it doesn't happen. In interkinesis phase, S phase is absent or DNA replication doesn't happen. After interkinesis, meiosis 2 takes place, and this meiosis 2 is equivalent to mitosis and it has 4 phases: prophase, metaphase, anaphase and telophase. The only difference between the mitosis and meiosis 1 and 2 is that during anaphase, the separation of two sister chromatids occur and the second sister chromatid has the part of the exchange chromatin material from the homologous pair. The meiosis 2 has 4 phases: prophase 2, metaphase 2, anaphase 2 and telophase 2. All these phases are equivalent to mitosis. In prophase 1, there the dissolution of the nuclear envelope occurs and the condensation of the homologous chromosomes occur, and then the nucleoli disappears. In the metaphase 2 stage, the homologous chromosomes get aligned on the metaphase plate, and that occurs due to the binding of kinetochore microtubules to them. In anaphase 2 stage, the sister chromatids of one homologous chromosome moves to two opposite poles and that is due to the shortening of the kinetochore microtubule. At the end of the telophase, there is the formation of 4 cells having the single sister chromatid, and the nuclear envelope again forms and the nucleoli appears in the nuclear region. We know that during meiosis 1, the 2 daughter cells were formed, and the same process occurs with the second cell in the prophase 2, metaphase 2, anaphase 2, and at the end of telophase 2, the 4 daughter cells are formed, the 2 from the first cell and the 2 from the second cell, where the 2 daughter cells have the maternal or paternal homologous chromatids, and the rest 2 has the combination of the first and the second maternal or paternal chromatids, or they have the exchange part of the maternal or paternal chromatid. Thus, at the end of telophase 2 or end of meiosis, 4 daughter cells having 2 the paternal chromatids and the 2 having the recombinant chromosomal materials are formed, and all these cells are having just half the chromatid material or the chromosome number than to the mother cell.